extend a warm welcome to you and share a little bit about what we're about and why we would invite Praveen Sethupathy to be with us. Our mission here at Upper House is to curate experiences of Christian thinking, being, and doing. So we do that by engaging our university, campus ministries, local churches, and the broader community, seeking to ignite theological exploration that enriches the life of the mind, striving to cultivate a virtuous sense of Christian identity, and working to embody a faith that has a positive and transformative impact in the world today. We usually do the work that we do in partnership with others, and this example is no different. We're thankful for the UW Faculty Staff Fellowship, who have helped promote this event early in the semester, and as you can see, the turnout suggests they did a fine job. And especially to Blackhawk Church, who originally extended the invitation to Praveen to be a part of their services yesterday, and he was generous enough to stay on for another day to be with us, so thank you to Blackhawk. I also want to note, for those of you, especially the scholarly among you, our affiliation with the Consortium of Christian Study Centers. So Upper House is affiliated with this a national organization that partners with 30 other campus groups that are working around the country to give voice to and advance the Christian scholarly tradition, all in their own way. And here at Upper House, you'll see on your events calendar that we have placed at the tables a variety of speakers coming up this fall that we're very excited about, ranging from legal scholars and historians like Samuel Moyne to university professors like Michael Lindsay and others who you may be interested to come see. So all of this serves our mission in a variety of ways, and we're delighted you're here with us today. Also, in that area of scholarly development, we have some exciting things coming up this year that we will be sharing more about as the year develops. For now, I just want to thank Praveen again for being with us. For those of you who are here this morning, you heard a very enriching conversation about the harmonious tradition between faith and science that Praveen argued for and articulated so well. To introduce him formally today is a friend of ours, a friend of Upper House and a friend of mine. The chair of the Integrative Biology Department, Jeff Harden, will introduce Praveen.
Well, thank you, Robert. I'd like to continue the list of people to thank by thanking Upper House for hosting these kinds of thoughtful events. And, you know, I'm just continually amazed at the remarkable meeting space that we have the privilege of gathering in. So thanks to John and Robert and Melissa and everyone else at Upper House for making these kinds of events possible. What is my privilege to introduce our presenter for today, and that's Dr. Praveen Sethupathy. Some of you who are here this morning will already know what kind of person Praveen is, but in case you don't know about his background, it's my job to fill you in a bit. Praveen grew up in upstate New York. He did his undergraduate work in Ithaca at Cornell in computer science, and then he went on to do a Ph.D. in genomics and computational biology at Penn. Following his Ph.D., he did postdoctoral work at the National Institutes of Health with Dr. Francis Collins. Many of you may know who he is. He's my boss, I guess Praveen's boss too, for that matter, in terms of my funding. But he's also a wonderful Christian man, and Praveen, I think, extends that tradition that you may know about from Francis, as you'll see today. Praveen is. I thought about talking about the details of some of the things that he does involving things called non-coding RNAs, but then I thought, no, I'd better not do that. He's a geneticist who is interested in molecules, and his research involves work that's funded by the National Institutes of Health, a number of other private foundations, and focuses on applying these kinds of technologies and this kind of thinking to issues related to human health. He's widely published. He is really a rising star in his discipline. He was selected by Genome Technology as one of the nation's top 25 rising professors in genomics in 2011, and that trajectory has continued into 2018. I got to know Praveen personally through his work with the BioLogos Foundation. I serve as the chairman of the board of that organization, and Praveen was kind enough to join our board, and it has been a delight to get to know him and to consider him a friend. Some of you may have also seen Praveen's talks through his work with the Veritas Forum. He has an incredibly compelling personal story about his Christian faith and about how science fits into that. And I'm guessing we'll hear a bit about that today in our lunch talk. Praveen is married to Rebecca Beck and has 3 children. Please help me welcome Praveen, and editing our DNA, exciting possibilities and important ethical implications. It's an important topic, and I can't think of anyone better to introduce this topic to us. So please help me welcome Praveen. Praveen, I have the dubious distinction of also awarding you your honorarium for today's talk.
Actually, there is another honorarium. All right. Very nice.
All right.
Very nice.
I'm sure you will wear that with pride when you go back to Ithaca.
Absolutely, I will.
Praveen, thanks.
Thank you so much, Jeff. Appreciate it. Thank you very much, Jeff. Very kind introduction. Don't feel it is deserved, but I will do what I can to try to live up to that. Many of you, or some of you at least, were in attendance for the lecture earlier this morning, and then maybe some of you are Blackhawkers who were there at the service yesterday. And I want to say thank you to all of you who listened, also came up and introduced yourself and talked to me. I met some fantastic people over the course of the last couple of days and have had very, very stimulating and encouraging conversations. So I appreciate that. I appreciate the feedback that you've provided in all sorts of ways. Those are important to me as I go on and try to engage and connect with other communities. So thank you very much. And I'm looking forward to this event as well. I'm thinking about this event a little bit differently. If you were in attendance with the other events, I had prepared notes that I walked through in the form of a lecture. I have notes and specific things I want to go through here as well. But what I thought is I might limit my own talking if I have a limited set of notes so that we can get into the Q&A time and hear from you, the kinds of things that you're thinking about. This is a very diverse crowd. We've got lots of different kinds of people, different training backgrounds, and so you're coming into this lunch conversation with ideas, questions, concerns of your own, and so I'd rather get to some of those and see if we might be able to wrestle with those together. I can't promise I'll have pat answers for you, but it would be great to hear from you and to have a chance to have a conversation. Does that sound okay? Okay. So as you saw the title, this is about genome editing. How many people have heard about genome editing? I'm assuming it's going to be most, if not all of you. Okay. And then how many people have heard of CRISPR-Cas9? Okay, great. So almost all of the people that raised their hands for genome editing had heard about CRISPR-Cas9. How many of you would feel comfortable explaining to one of your colleagues or peers or friends what CRISPR-Cas9 is? There you go. The one person, in the room who has more expertise than I do on this topic, and maybe another person over there. Okay, great. So I'm glad to see that. But this is what the kind of feedback I usually get when I ask this question is everybody's heard about it. We have some kind of an understanding about what it is, but we wouldn't feel comfortable articulating it, and maybe that makes us feel even more uncomfortable about how we feel about it, what it even means to us, right? And so whether it's NPR or whatever your news source is, where you're hearing about this kind of in your day-to-day life, I'm betting that most of you would benefit or would appreciate a basic understanding of CRISPR-Cas9 before we even get into what this talk is billed as, which is discussing ethical implications of this. And I find that that's really important, whether we're talking about evolution or stem cells or genome editing. So much of the consternation that some communities feel come from the fact that they're not even well-defined terms. People are using them in different ways, meaning different things, and therefore speaking past each other. So I'd like to avoid that in as much as is possible today, and start with a brief description of it and a brief history of it so that you appreciate kind of where we find ourselves today. And then we can get into the meat of it, okay? So CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. Okay, that's what CRISPR stands for. Whenever I speak on this, I always make sure it's in front of me, okay? Ever since Craig Mello, a Nobel laureate, was giving an interview for NPR, and live was asked, can you tell us what CRISPR stands for, by the way? And he couldn't come up with it, which is not to malign Craig, because I can't either. I often forget, wait, interspaced, regular, so don't feel bad if even walking out of here today you don't remember exactly what it is. But it is clustered, regularly interspaced, short, palindromic repeats. Okay? What does this mean? Okay? Let's take a step back and talk about DNA. Right? DNA present in almost every cell of our body, and it's made up of these chemical bases, A, C, T, and G, right? And we have stretches of this from 1 to 3 billion in every cell of our body that has DNA, right? And in different stretches, you can have what are known as repetitive DNA sequences. So imagine a pattern like ACGGT, okay? And then imagine replicating that pattern over and over again. So you have ACGGT, ACGGT, ACGGT in this kind of clustered sequential fashion, okay? That would often be called by geneticists or genomicists as repetitive sequence. And there's a lot of repetitive sequence in DNA. That in and of itself is a field. You can start up a lab and study for the next 5-6 decades about different kinds of repetitive sequences in the genome and what they mean. And frankly, it all started when we were sequencing the human genome and realizing that these repetitive sequences were far more frequent than we had even previously anticipated, even though we did know that they exist. Right? And people are still studying many different kinds of repetitive sequence. It's not just one pattern that's repeated, but it's different kinds of patterns. Maybe it's GA, GG, GAG, and then that gets repeated over and over again. The fact that it's repeated that way is some sort of a clue that perhaps the clustering of the same kind of pattern means something important, is doing something functionally important. That's the basic idea. Okay? But these repeat sequences, the CRISPRs, were first found in bacteria. So it was actually 1987, and it was a lab in Osaka University, where they first identified these repeat sequences in a bacterial genome. So not in the human genome or mouse or fruit fly or anything like that, but it was in bacteria. Right? And I always really like to point that out because sometimes people ask, why study bacteria? Outside of the context of bacteria infecting us, who cares? Right? There's so many fundamental discoveries that have affected human biology and human disease that we now take for granted that come from studying things like fungi and bacteria. Right? CRISPR-Cas9 being one of the most recent and popularly known. Okay? But these are repeat sequences that are found in, at that time, was one bacterial genome. We now appreciate that they're found in many, many bacterial genomes, and archaea and other kinds of species as well. But after it was found in 1987, it was found again in some other bacterial genomes in 1993 by labs in Netherlands and Spain. So things were starting to pick up. It wasn't just some weird anomaly that was being found in one bacteria. More and more people were finding these repeat sequences in other bacteria, right? And so there was a growing interest in what these repeat sequences were doing. It's one thing to find something in the genome that you didn't know about before. It's quite another to try to figure out what they're actually doing, okay? So what does it mean to say that they're interspaced repeats? It occurs to me here that a quick slide might have been helpful, but hang with me because I'll try to describe for you how it's arranged, okay? So imagine my fist as one of those repeat sequences, okay? The next repeat sequence among the cluster is not found immediately adjacent to it. It's found right about here. It's interspaced, right? And then the next one isn't right here. It's right about there. and it's roughly equally spaced in the cluster that you find it in the genome. So what's the stuff in between, right? The stuff in between is also DNA, okay? It's in the bacterial genome, but it's not the repetitive sequence, it's something else altogether. It turns out there's information encoded in that stuff in between that allows the bacteria to be able to fight against foreign invaders. And I need to explain this for a second, because usually we think of bacteria as our foreign invaders, right? And they are, but bacteria themselves have foreign invaders, and they're called bacteriophages. Right? And they are essentially viruses that infect bacteria. So we're very familiar with viruses that infect us, but there are different kinds of viruses that specifically infect bacteria. And it's this dynamic between bacteriophages and bacteria that prevent one or the other from taking over the world, okay? Because if we didn't have bacteriophages, we'd have way more bacteria than we knew what to do with, right? It'd be hard to even imagine us existing, right? So that interplay and dynamic is extremely crucial. But you can imagine that the bacteria have developed ways in which to fight against these bacteriophages that infect bacteria. Okay. A quick aside here, is anybody, we use the word bacteria and bacteriophages, particularly in the scientific community, a lot, but does anybody know where those words come from, what they actually mean? Aside from Jeff. Probably some others here as well. But bacteria, okay, comes from a Greek word bactron, okay, and bactron is actually a staff, right? It's like a staff or a stick. And bacterion is like a little staff or a little stick, right? And the reason why bacteria were named that way is that the first ones that were discovered were kind of rod-shaped, like a rod or a staff, right? And so when scientists noticed that, they gave it the name bacterion, right? And then bacterium in Latin, and then bacteria today, okay? And a phage is essentially, again from Greek, but it means to devour, right? To like engulf and destroy, right? So a bacteriophage is essentially something that can infect and devour a bacteria, right? And so that sounds pretty nasty. And so you can imagine that the bacteria want to mount some kind of an immune response against these foreign invaders, the same way that we expect our body to when we ingest pathogens that are not so healthy for us, or get infected and get the flu and things like that, right? Okay, so makes sense that bacteria need a way to respond to foreign invaders. It turns out it was not until around 2007, so this is, we're 20 years after the original discovery of these oddly, interspaced, repetitive palindromic repeats that were discovered, that it was finally proposed that these are probably an immune response, that the information that's encoded in between those repeat sequences actually helps the bacteria to identify, target, and kill and disable the bacteriophages, the viruses that are infecting that bacteria, okay? So it turns out it's a really clever bacterial trick, right? It's a way, it's something, a mechanism that it's evolved to fight invaders, right? So that's where it originally comes from. It's not the context in which you usually hear it. But again, as I said, I find it helpful to understand the history and how sort of this all came about, okay? It wasn't really until 2013 or so, about 5 years ago. Feels like genome editing and CRISPR-Cas9 has been around forever, right? But it really wasn't until 2013 that that mechanism of bacteria, scientists began to realize we could leverage that. We can take bits and pieces of that and use it for the purpose of genome editing, right? The idea was nature has already come up with a way to fight foreign invaders and disable invading DNA. What if we used some of that same machinery to make changes in human DNA, right? In cells in a dish, for example, right? And so that was done in 2013. We were starting to use this technology to edit the DNA of cells that were being grown in a dish, which is a very, very common occurrence in most molecular biology and cell biology labs in the country. Okay? And it's now 5 years later, And it feels as though it's been around for several decades because almost every major molecular or cell biology lab that has funding is either doing CRISPR in some way or thinking about doing CRISPR or collaborating with someone doing CRISPR, right? And so even though it took 20 years to figure out what these were even doing in bacteria, we have now very rapidly grown from that point to be able to leverage that machinery for things that it was not originally intended for, right? But as a tool in research. Okay, so I hope that background is a little bit, it's helpful for you now and in future discussions when you hear about CRISPR-Cas9 and genome editing. It wasn't something that was discovered from scratch. It wasn't machinery that was generated and assembled from square one. We were actually leveraging something that was already happening in bacteria. Okay, so with some of that background out of the way, why is there so much buzz about CRISPR-Cas9? What is it? What are we using it for now, and why is there so much excitement and buzz about it? Why is it always on NPR, for example? Right? Basically, all right, as I've already alluded to, it's a method for genetic engineering. Right? And this is sort of a broad term that's used to describe a process by which you alter the genetic code of an organism. Right? So you can think about genetic engineering in plants, you can think about genetic engineering in mice and fruit fly and model organisms like that in the laboratory setting, and you can think about it with humans. Right? And we're going to get to that. Right? But there are all sorts of different kinds of contexts in which genetic engineering comes to play. And this is a method for genetic engineering. But that's not new. Genetic engineering has been happening for decades. Right? What makes this exciting is not that it makes genetic engineering possible, it's that it makes it much, much more accurate than ever before, and much easier to use, and much more rapid to execute, and much more cheap fashion, right? It's like a democratization of genetic engineering. Right? You know, in the past, the tools and the resources and the technologies for genetic engineering might only have been available to a smaller set of laboratories that were really focused on that kind of work, and perhaps it was not as accurate and maybe not as cheap. And for various reasons, this particular technology is a lot more accurate, a lot cheaper, a lot faster to do, which is why it's easier for everybody to adopt, right? Well-funded labs, not so well-funded labs, people with expertise in molecular genetics, not so much expertise in molecular genetics, can still get the reagents, the tools that are necessary to pull this off in their context of interest in their lab. That has really what has made this really exciting, is it's just spread like wildfire in a way that previous technologies, due to various limitations, were not able to. Right? So now it's on everyone's lips. Right? And that's making it more of a reality when we talk about things like, oh my gosh, is this going to come into the human realm? And if so, what are the ethical considerations? Now that's getting hyped up as a point of conversation more than with all of the other previous technologies. So that's the thing to understand about it too. Right? It's not necessarily the first method for genetic engineering. So as a research tool, we are leveraging CRISPR-Cas9 in my laboratory. Okay? And we're using it in some very, very exciting ways. And so I'll give you one example, just as a quick story. One of the diseases that we study in my laboratory is called fibrolamellar carcinoma. It is a very rare, very devastating form of liver cancer. Until recently, it was not even often properly diagnosed by clinicians who would see a mass on the liver and think that it was a typical liver cancer. But it turns out that in some cases, it's actually this rare form which doesn't respond to any of the drugs that have been developed for people with a typical liver cancer. And the nefarious thing about this disease is that it affects little kids to young adults, and the survival rate is not high. And because we have no treatment option right now, these children and young adults essentially die 5 or 6 years later. On average. There are some amazing, almost miraculous success stories with a variety of drugs that have been attempted, but by and large, people die. And it's really devastating. I've met with these communities and their parents who, these happy, healthy kids, and then suddenly, they have tummy aches for some years, and it's not clear exactly what's happening, and then before they even think to see an oncologist, this cancer has spread throughout the body. So it's a slow-growing cancer that doesn't tell you it's there for a long time, and then before you know it, it spreads like that. Right? These are these two features of cancers: how fast they grow and how fast they spread. And this particular one grows slowly but spreads quickly once it gets to a certain maturity. So it's really devastating, and we've been working hard for the past 4 or 5 years since we learned about it to try to come up with a cure for these patients and for these families. And for various reasons that I won't get into, it turns out to be a disease that really lends itself well, potentially, to CRISPR-Cas9-based treatment. Right now, we're not doing anything with human patients or anything like that, but it's a single mutation that causes this cancer in these patients. And that's an important point because a lot of cancer is not that way. It's an accumulation of many mutations, right? And so you have colon cancer and you go to your doctor, he or she might tell you, it's because you've got these 7 or 8 or 10 or 12 mutations that in accumulation are increasing your risk for the development of colon cancer because of the way you eat, right? It's usually very complicated interaction between your lifestyle and your diet and your genetics, many mutations potentially contributing. But here's a cancer where it's one mutation. And if we could fix that, if we could repair that one mutation in just the small collection of cells that originally have it in these patients, we might be able to reverse course, right? And so when we say exciting possibilities, right, I'm getting goosebumps talking about it because I'm flooded with the thoughts of these patients in my mind, right? And we have this amazing opportunity to give these people a life that they're not going to have otherwise, right? Right now, our efforts are limited to cells. So we have cells in the lab that are able to mimic how this cancer behaves, right? So even the discovery of that was really big for this field. But now that we have that, we call it in the field a model, right? So it's a model of how this cancer behaves inside of a person. And we have that growing in Petri dishes now, right? And now we're able to test our ability to be able to use this CRISPR-Cas9 technology leveraged from bacteria to see if we might be able to change that, reverse that mutation in these cells that are harboring that mutation, and see if we can stop it from behaving like a cancer. That's what we're doing right now, and we've got a long way to go, but there are promising preliminary results, right? So I'm hoping that in the years to come, I'll be able to tell you even more about this and have success stories to share. That would be my dream. Right now, we're in early stages where we're just trying to figure out whether we can even do it, right? But we've never before been as excited as this because we have the technology to do it, and it's not going to cost an arm and a leg as it might have before, right? So that kind of thing is being done not only with cancer, but other kinds of diseases. But for the most part, in most labs across the country, it's happening in cells, or it's happening in fruit flies, or zebrafish, or mice, the kinds of species that are generally used in laboratory settings in order to try and rectify human diseases or understand why they come about. The use of these organisms is not new. That's been going on for a long time. And the use of these organisms to screen different kinds of drugs for human diseases is not new. All that's new here is that this CRISPR-Cas9 technology is now being tried out on these different models. And so that's rampant. It's going on all across the country and the world. And generally speaking, there are no really robust ethical concerns to those. I mean, to everything we do, there might be some ethical concern, but those aren't the ones that I think gain the most traction when we talk about ethical concerns with CRISPR-Cas9, right? Though that's leveraging a technology, studying it in model organisms in the hope that we might better understand our biology and rectify our diseases. Okay, so that's happening a lot. Okay, where it starts to get sticky is in thinking about CRISPR-Cas9 as a therapeutic intervention. Okay, that's the first layer of stickiness. It's not the most layer, it's like the first layer of stickiness, right? Is CRISPR-Cas9 as a therapeutic intervention. As you can see, my mind is already going that direction, right? Because I'm hoping to provide relief for these patients. And so you can tell if my mind is going there, many, many other people are already there. And in fact, it's already happened, right? So we need to stop talking about this as a future tense, as something that can happen in the next 5-10 years. It's already happened. In 2016, China has been, labs in China, I should say, are trying to leverage CRISPR-Cas9 in humans to rectify cancer, so metastatic lung cancer, for example, or other kinds of cancers, but that was the first one that was attempted. And a clinical trial has now been approved in the US, led by the University of Pennsylvania, where I did my graduate studies, and a few other institutions I think are involved as well. Okay, so this is happening right now, where they're trying to see if this could be useful for patients that are suffering with certain kinds of cancers. Okay, cancer has been the primary focus right now, but the application of this, should these clinical trials be successful, to other kinds of diseases for which we know specific mutations to cause the disease, is just on the horizon. And in fact, they may already be happening and I'm not even aware of it, right? So this is ongoing right now. What are the concerns? What are the concerns that are associated with trying to do this in humans? When we talk about doing this in humans, there are two different ways, broadly speaking, in which it might be applied to humans. One we would call is rectifying somatic mutations. The other is rectifying germline mutations. And they're two different conversations. And I'll tell you what the difference is. With somatic mutations, these are not mutations that you inherited from your mother or father. Okay? These are mutations that are new in you. Right? So if you bask in UV rays too much and you get a mutation because of that exposure and then you get melanoma, those are somatic mutations, mutations that occurred in cells of your body that are not germline cells, sperm or egg. Okay? What that means, that the reason why I draw a distinction between that and germline is because you don't pass those on, right? Unless the mutations occur in your sperm or egg, or what's called germline. Okay, if they're somatic mutations, you're not passing them on, so they're unique to you. Okay, they're just as ravaging and devastating and can cause horrible things like cancer, but the difference is that you don't pass those types of mutations along. Okay, if you catch certain kinds of cancers early enough, it's just a small collection of cells that have this maybe 1 or 2 mutations that trigger what's called a transformation, or the development of the cancer. What if you could catch it at that stage and use CRISPR-Cas9 to reverse those mutations, to edit them back, right? Now you no longer have this small cluster of cells that is going to wreak havoc in your body, right? So coupled with the ability to detect it early, we may be able to leverage CRISPR-Cas9 to reverse course for these patients, or maybe they'll never develop the tumor at all, okay? That's the hope that, for example, this University of Pennsylvania-led trial has. Can we use CRISPR-Cas9 to do this? It's still a question. We don't have really good data on this yet, right? We've been doing it mostly as proof of principle in other kinds of settings, right? Now they're moving it to clinical trials. And the question here becomes, with these somatic mutations, are we comfortable with that? Is there any problem, should we have any ethical consideration with trying to rectify these mutations in these patients? Okay? There are some concerns to go over. Some are scientific, some are more ethically oriented. The scientific ones, is it even ready for prime time? You know, we talk about these things as though it's a panacea, but the reality is there are problems. There are what are called off-target effects. So you can custom design that machinery to target the mutation you want, but every once in a while it'll go somewhere else, right? And then if it goes to that somewhere else and introduces a new mutation, well, you've just corrected one problem and potentially introduced another. Are we okay with this? Right? Well, my answer to this depends largely on how good we are at this moment at mitigating those effects. And I'm telling you, we're getting better and better with every passing month and year. We're not perfect. And anybody that suggests that isn't being faithful to where we are. But we are definitely getting better and better, and it's the best that we've ever had for this kind of technology. Right? And the other thing I would say is, this is largely true for most other medications. And there's a difference in that we're changing the DNA, and other medications generally are not. But when we take a pill for our cancer or our diabetes or our schizophrenia or whatever it might be, right, we are familiar, very familiar with the litany of potential off-target effects on all of these medications, right? So I am not necessarily saying it's the same thing, but it is helpful to put it into the context of what we're already doing and generally comfortable with. The clincher on this is that at the moment, it's being proposed primarily for patients who have no other recourse and are likely to die very soon, right? There are different names given to this. One that I'm familiar with in the circles that I'm in is a mercy protocol. This is a protocol that the FDA will approve quickly for a patient that doesn't have very long based on clinical expertise, but you have some drugs that you might want to try just in case it works, but it hasn't really been proven yet. And if the patient accepts it, it can be done. So again, these kinds of things are already happening, right? So it seems to me that's, in my opinion, the least controversial or problematic, right? Yes, there are some issues to work through here, but they seem like workable issues, and I think by and large, the desire to alleviate the suffering of these patients is going to overwhelm whatever other kind of subtle problem we might have with these, okay? Now let's get to the second track of the kinds of mutations we might want to rectify. These are germline mutations, right? So not the mutations that are occurring in other parts of our body over the course of our lives, but in our sperm or our egg, right? Suggesting that changes made there are heritable. We're going to pass them on to the next generation. And here's where things start to get a little bit fuzzier, right, in terms of the ethics. Right? Are we comfortable with the longer-term changes? Right? There may very well be some complicated kind of long-term changes of taking medications that we take today, okay? So you still have that analogy, but it starts to break down a little bit because you're making a very definitive change in the DNA that is then going to be inherited by the next generation. Right? What could be the potential problem of that? Well, you might be convinced in this patient, with the rest of his or her DNA, making this change is okay. But what if that change is made in the context of someone else's DNA? Could it have a worse impact? Could there be some unintended negative impact, right? It's just difficult to map out what the consequences are of spreading this in the human population, right? Of course, mutations like that occur naturally all the time. So it's not like that doesn't happen. The difference here is we're introducing it, and the question becomes, are we comfortable doing that, right? That, too, has already been done. So in 2017, at Oregon Health Sciences University, for a disease called hypertrophic cardiomyopathy, right? And we don't need to get into the details of this except to say that it's very devastating. It can cause sudden heart failure and death, instant death. Is a very viable outcome for people that have the mutation that causes this disease, right? So it's heartbreaking, and it's unfortunately not as rare as I would like to see it be, okay? So it's already happened. The early stage zygote, so this is a fertilized egg, right, can be altered at the genetic level using CRISPR-Cas9 technology to revert this mutation back to the protective version, right? So get it out of the risk version and get it back into the protective version. So Oregon Health Sciences University received a lot of press about a year ago for accomplishing this and publishing it in a scientific journal. So the concern here is, are we starting to play God? Right? Are we playing God is a question that is often levied against scientists that are involved in this kind of work. Okay? And there are a couple ways in which I want us to kind of think about this. Right? The first is, I want you to be aware that this question of are we playing God has been asked throughout history with every new wave of technological advancement. And it's important to understand the context. That doesn't mean, don't get from this that I'm saying there's no new question to be asked in this particular technology, but it is useful to put it into that historical context, right? In fact, you know, in terms of Christian history, right, there have been times where with the development of and more widespread use of medicine, that Christian communities have rejected that in favor of God just healing us, right? Or you know, if suffering comes about, then it must be for our advancement, like Job, right? Or if you know, God wants me to die, then perhaps that's the best course instead of me trying to resist what He's doing in my life. These are not fringe. They may today be fringe statements, but at various times in history they were not fringe. Right? They were endorsed by the power structures in the organized religion and were practiced by many people who were Christians, Catholics, related faith traditions. So you know, there's a prominent example. Saint Gorgina is, I think, her name's a Greek Orthodox saint, and I think she's the patron saint for people with illnesses in that tradition. And the story goes, I think she was about 4th century, and the story goes that she was trampled by mules and had crushed bones and crushed internal organs, and because it was indecent to receive medical treatment, you know, she is said to have not received any treatment and simply prayed for miraculous healing, which, as the story goes, was provided for her, and she was elevated as a saint in that community, right? Because the notion of resisting external influences to our body and accepting whatever it is that God is doing was a dominant feeling. That was what was thought was orthodox and proper and decent and right thing for a Christian to do in that community, right? So you know, we don't have to even go that far back. I mean, even 17th, 18th century, there are resistance movements within Christianity that say, you know what, if God is bringing suffering, then, you know, perhaps I'm supposed to be going through this, you know, that we shouldn't be looking for natural means by which to ameliorate this, because maybe I am then foregoing some sort of spiritual growth, right? Again, not particularly fringe. I also don't want to give the impression that every Christian believed this. That's also not the case, okay? But these kinds of ideas are much more prevalent than we are used to thinking about today. But what happened over time? We find ourselves today saying, if you've got a headache, it might be a good idea to help you out and take something. If you've got bipolar disease or, you know, OCD, it'd be helpful for you to take some medication and give yourself a little bit of buffer space to work through that. You know, if you've got diabetes, you name it, right? We're generally in the Christian community not opposed to the notion of taking medications to help ourselves out. And in fact, the conversation has shifted to another kind of ethic where we say, no, we have to alleviate suffering. We have to ameliorate pain. This has to be a part of what we do as believers, as Christians. You know, being the hands and feet of Christ to a broken world, we need to, this is a part of that. We have medical missions now, right? That's a 180, right? So it's useful to think about that, right? And say, okay, is all of our hype up about the concern about this, you know, is there part of it at least that I might be able to kind of calm down about, right? Is it just the next iteration of medications and drugs? Or the next iteration of, advanced surgeries? You know, the ways in which we've responded to them in the past. So I think that's useful to think about as you are working through how you feel about this, okay? But on the other side, there are a couple of things to think about, okay? The first is, in order for us to get to the point where we feel that we have leveraged the technology well enough
to be able to put it into clinical trials or use in humans, it requires a lot of research work. A lot of that research work, in order to be as relevant for humans as possible, has to happen in human embryos. Right? And a lot of the times we'll get it wrong as we try to make it more perfect. Right? What happens to all of those human embryos that are utilized in the research process? Generally speaking, there isn't a problem that most researchers feel that are engaged in this topic in just destroying those. Right? So now we've just introduced the other side of the coin. Say, well, wait a minute, we've been talking a lot about alleviating suffering, and I'm on board, right? But now we're alleviating suffering at the expense of what many of us might say is another human life, or at least a life with potential, right? So then, how do we feel about that? Right? Earlier this morning, we were sort of half joking when we said that you need a tension between science and faith. There's harmony, but think of what harmony actually is. It's not the same notes, right? There's a little bit of a tension there, right? But it's a tension that works well together, right? And it's the same thing between science and faith. We can't go so overboard with our desire to want to heal that we have no ethical foundation, right? And the example I gave was, you may have a failing kidney and I really, really want to help you, but I can't just rip it out of him to give it to you, right? Then what have I done here? I've really lost all ethical bases, and I'm not really moored in anything, And so that's where we come to with this question. It's, yes, we want to alleviate suffering, but it seems like to get there, we may need to make some compromises with things that traditionally we have been very uncomfortable with, right? So that's a line that, I'm not sure how to navigate that, right? Instinctively, because of the caution and the slowness that I think we need to apply to any kind of new progress. And that's maybe the most important word for today, by the way, right? It's not about stopping something or not doing something, but it's about applying caution. We live in this age where we thought of an idea, we want it to happen yesterday, And I think we need to slow things down and give ourselves the room to think about it, right? But as I slow my thoughts down on CRISPR-Cas9, this is one big area that gives me room for pause. Am I comfortable with this in order to achieve that? And I'm not sure yet, right? And I respect a lot of Christian voices on both sides of the answers to that question. I've heard yeses and nos, but they basically boil down to how they weigh these things, right? Whether they believe this matters more than this. And I think that is very difficult, but a starting point has to be, what is your motivation? Right? What is your motivation for doing? Why do you want to do this? And I'm not saying that is going to, in and of itself, solve the problem, but it will at least get us talking about the right thing. Is this motivation really what we ought to be focused on in the Christian body? Right? The more salient concerns that you've heard about, not that isn't important, but it's a well-defined question. The kind of universe of other kinds of scarier possibilities are things that you may have heard about in the kind of general public conversation of this, like the use of CRISPR-Cas9 for designer babies, and the use of CRISPR-Cas9 for biological warfare. Right? These are all contexts that make this technology very scary. Right? So designer babies being the idea of personalized enhancement. Right? You want a kid who's blonde and blue-eyed, and let's make it happen with CRISPR-Cas9. Right? This technology gets into the wrong hands. Maybe it can be used, for nefarious purposes. Right, in the same way that other kinds of biological weapons are, right, like viruses and things like that, right? So those are very, very, very important concerns, because if we go too fast, right, we will not, even if we decide we want to move forward and do this, if we don't do it cautiously and slowly, what we will not have put in accountability structures, regulations, things like that, in order to severely mitigate or minimize the probability of these kinds of horrible things, right? And again, we've done that kind of thing before, right? If you're going to work with Ebola, you got to jump through so many hoops to get to the point where you're going to do that, right? And the purpose of that is to restrict access to things that could otherwise, in the wrong hands, wreak havoc in our communities, right? So you could, in principle, imagine doing things like that with CRISPR-Cas9, should it be something we move forward with. But I firmly believe that if we don't take it slow and walk through each of these things, no matter how obvious it may seem to others, we won't actually put in place the best structures of accountability and regulation. So that's one reason in and of itself to take it very slow. And I'm heartened to hear, it's not just Christians talking about this, many non-Christian scientists are coming to the table in a way that I haven't seen before with other kinds of technologies that elicited these conversations and saying, whoa, I'm not saying let's not do it, I'm excited about this, but let's talk about this. Let's bring the key players together and talk through this. And so if there's a silver lining or a ray of hope, it's that many of my colleagues and many leading scientists in this area, not all, but many leading scientists agree with the importance of taking things slow. Right? But we can't just dismiss things like designer babies, personalized enhancement, as, oh, yeah, is that really a big concern? Right? There are already people talking about these kinds of things. Right? And while it may not be happening right now, or while we may still need more development for people who want to apply it in those kinds of ways, provided there isn't regulation. And it's not just America, right, guys? I mean, we can have regulation, but there are countries in the world that are plowing forward much faster than we are, I think ultimately to the detriment of this field and its correct application of the technology, right? So there's a worldwide enterprise to think about, not just what we should be doing as a country. And so we need to be engaging with other countries and drawing them into this discussion rather than alienating these countries and then making it difficult for us to have joint efforts, international efforts about how we pursue these things, right? And I'm not a Pollyanna. I'm not suggesting it's all going to be easy if we just play nice and hold hands and sing Kumbaya. But we do need to take this effort because it's very serious. This is stuff that's on the horizon, right? And we have to remember when we think about Nazis, when we think about eugenics movements, this wasn't unique to Nazis in Germany. There was a robust American eugenics movement, right? There were times, even apart from eugenics movement in our country, led by scientists, right? There were times in our country where cells were taken from people without their consent. Sorry, consent, yes. And many other examples like that where we have forayed into realms that are clearly unethical and inappropriate and strip people of their dignity, right? And that should concern us to the core of our being, right? Because we are not immune to this. It would be erroneous to think that we already have everything in place to prevent this. We don't. And that's why this conversation is extremely important, because we have already revealed that brokenness and that wretchedness and that sinfulness in us, that tendency to take this in directions that clearly strip people of their dignity. And with every single person being made in the image of God, being an image bearer of God, that should seriously offend us as Christians, right? We can't take that lightly, right? And so we need to be at the forefront of providing this voice. And if others are joining us in this voice, fantastic. But we need to be there making this voice heard irrespective of who else is there, because this really gets to the heart of what I think is perhaps the greatest concern down the road in the application of this technology. Okay? So what I'll end with is something I alluded to before, and that is that with these kinds of questions that emerge from the scientific community, and they start to, make us question about the relative merit, whether or not we should engage it, how do we view it as Christians, I hope that it's an encouragement to you that the starting point is not, what's the technology? Because it's not usually the technology is bad or the technology is good, right? I think it was Jeff Hardin in an article in a lecture that he gave where he pointed out the words of Billy Graham, who said, it's rarely the technology, and I'm paraphrasing here, but it's not so much the technology Christians ought to be worried about, it's the people using it, right? And my interpretation of that is, what is our motivation, right? If we can all come to the table and agree on a set of motivations that we believe outweigh any other concerns we might have in terms of its application, then we can lay out very, very clear lines that ought not to be crossed with clear regulation and clear penalty, right, for crossing those lines, right? And a body that is dedicated to the application of those rules. None of those really exist right now, right? And so we need to sit down and put all of that together. But I think the starting point is, why do we want to do this? You know, because we can is not a good answer. And it's surprising how often I do hear that, right? So that's why our voice here is important, the Christian voice. It's not because we can is not a good answer to me, right? We need to have articulated and formulated a much more compelling reason that is consistent with our purpose as people made in the image of God. We are called to reflect and represent him in this broken world, right? And so is my motivation consistent with that? And even there, we can have honest disagreements as Christians, but again, like I said, at least we're talking about the right thing and then trying to form some kind of a compromise there, right? So that's what I would recommend is the starting point here, and not so much just, is our technology ready for prime time, or what are the off-target effects? These are all important concepts and topics to be discussing, but look, we're headed here, so let's get ahead of this and start talking about what our motivation is. Why do we want to do what we want to do? And then I think a lot of the other stuff will kind of sort itself out, and then we'll get really to the heart of the matter and discuss the toughest questions. So that's what I have prepared for today. Thank you for listening to me. I am happy to take any questions you have on any of this. And the hardest of the questions I will pass along to Jeff Harden. But honestly, anything else about my research or work in genetics or anything like that, I'm happy to field. I'm an open book.
Thank you, Praveen. So yes, we have plenty of time for questions. Either I or Melissa will go around and hand you the mic for the purposes of our recording. If you could speak into it, that would be great. And if you could construct your comments in the form of a brief question, that's always welcome.
Okay, first off, thanks for the presentation. It was really good. I loved how you broke out the different ethical dilemmas.
Great.
So one that's in my head, and let me try and articulate this briefly. We talk about government regulation and how it can help to limit how quickly we go about rolling out new technologies. Where my head goes to when I look at history, like Nazis, Soviet Russia, socialist countries, my fear is the opposite as well. Mm-hmm. How do we prevent the government from going too far? Mm-hmm. To say, oh, this is great, we can heal this cancer, let's pass a law that every baby has to get treated for this, because it's kind of like we're wrestling with monsters that we don't know where their head starts and their tail ends. Yeah. And what damage do we do as a human race by removing genes that we don't yet know all of the historic implications to why they were in our code to begin with?
Yeah, you articulated it very well. And I think that it is a veritable concern, right? By no means that I mean to suggest that private industry would run amok with this, but the government would be very moral and ethical. But not at all suggesting that. But there may be a couple of things. I don't have an answer for that. Okay, I'll be up front. But there maybe are a few thoughts. Right? The first is, again, this is why I think that people with a strong, well-defined foundation of what's good and what's not, and where that goodness comes from and how goodness is defined, right? So people with that kind of principle and foundation are important in every sector and realm of our society, right? So the problem that you're mentioning underscores the importance of that concept, right? And so we need people in every sector so that there is that pushback to these kinds of, a rise in this kind of thinking that could very well happen, right? So that's the first and most important reason why I even talk about this stuff, is that I just want more people to know about it, to understand it, and not to bring about hysteria, but to say, okay, some of it could be really good motivation, some of it I can already see it could be very bad motivation. I need to be better educated about this so that I'm holding my government responsible, so that I could maybe even work in sectors that control this. So that's why I think that these kinds of forums are very, very important. Right? Secondly, I think I would just say that as a corollary to that, very grateful for the country we live in, because although the nature of political discourse is increasingly volatile and sort of vitriolic, we still have this structure, I believe, perhaps naively, where we can, as people, enact change. Right? And so that's where I think American leadership is very important, even when it comes to these kinds of topics, because we have that opportunity to actually bring about change. And so we need to have information, and we need that information from people we trust so that we will then actually act on it. Right? So most churches in America, I mentioned this morning, scientific questions, 30% of evangelicals ask, according to a Pew poll, ask their pastor about scientific questions. And I can't imagine being this pastor, right? Being asked about CRISPR-Cas9, that's a huge burden. I mean, you don't know where to turn or how to address these questions, right? And so we need more people in these communities who are knowledgeable about these things, talking about it with one another, so that we're better informed and then able to push back on our government. I think that's got to be the way. And then, when people that are working in these sectors, in the National Institutes of Health and in the FDA, and that are able to kind of push back on things like this that might happen. And that's happened in the past, right? Sometimes, unfortunately, later than I would have wanted, but it has happened.
It seems like a lot of the questions related to this issue center on the question of what is the ontology of an embryo? So, like, what is an embryo, and should any and every embryo be treated with the same dignity and respect as a human being or not? It just feels like a lot turns on like that question. I was just wondering, is there right now a consensus in the Christian world on like the ontology of an embryo, or or maybe what's your view on that?
Great question. Thank you for it. I am not personally aware of a broad consensus, which is why I think this it engenders very strong emotions and sentiments. I kind of take the Jeff Hardin line on this, which I'm going to paraphrase, but if Jeff, I have mischaracterized your position, please jump in. But I have found his position to be very helpful, so I'll kind of channel that, because I think that's how I think about it as well. And that's that I don't really think that there is extremely clear language in the Bible that allows us to chapter verse the answer to the question of an embryo. Okay? And what I mean by that, right, is I can't just go somewhere that says God said embryos are something, right? So without that, what do? I try to make the best inferences that I can from my understanding of Scripture in a holistic manner. Right? And having attempted that and listened carefully to others who have attempted that, my recommendation would be to move with caution, because although I don't know exactly where God believes life starts, at which moment in time the image of God is conferred upon that entity, I don't know the answers to these questions, and I'm fairly certain I'm never going to know on this side of heaven. Right? That being said, because I don't know definitively, my instinct is to be as cautious as possible, right? And so what that means is to take it back as early as possible in the definition of what I think I would start to get very uncomfortable mucking around with, right? So I think that's the way that I would think about it. But it's pliable for me. That's one of those things where I think people of good faith and equal commitment to Christ can differ, in my opinion. I know fellow brothers and sisters who they will admit that it's arbitrary, but they just draw the line somewhere else, right? They say, the nervous system has to be developed to a certain point, right? I mean, I get that in terms of feeling pain, perhaps, but I don't know what that has to do with the image of God necessarily. And maybe God will correct me one day and say, it has everything to do with it, but I can't find anything in Scripture or in Revelation in my life that allows me to make that claim, and so I just end up being as cautious as possible about it, right? But I'll tell you this, okay, and it, you know, doesn't always go over well, but we have to make imperfect decisions all the time as a Christian. You know, I tell my kids, don't lie, right? But then I also teach them, you know, if you were, you know, in Germany or in Austria or something like that in the Nazi period, and there were Jewish people that you could help hide, and would you hide them and then lie to the German officer that you would? Like, yes, I forcefully teach them yes, right? So what am I saying there, right? That I'm saying there's context dependency to God's laws, and yes, I am, you know? That's scary for many people because then it's like, oh my gosh, like if we don't have immutable lines, then isn't this all gonna disintegrate and fall apart? And I think this is where daily walk with God is important. That thing that we're always talking about as Christians is constantly interfacing with God and trying to understand what the right thing is to do in this context. We're not going to be able to do that well as individuals or as a community if we're not cultivating that relationship. And it makes the answer ambiguous, nebulous, non-formulaic, but that turns out to be the Christian life in my experience.
Well said. Go ahead.
I'll start here, and then there's one over there.
Thank you very much.
You're welcome.
Informative and terrifying. Many folks here know I work in the environmental field, so I have lots of things that keep me up at night. But
Just added another one for you.
yeah. Well, I already knew a little bit about it, but the line that bothered me most in your entire thing was, CRISPR makes genetic engineering easier, more accurate, quicker, and cheaper. I have a lot of international background, grew up in Pakistan, happen to know that some of the smartest scientists in the world are in countries like Iran and Pakistan and other countries where the concept of regulation is, and we can add China to that probably, maybe even some labs in India, I don't know. You know, we've created the tool, and a little bit like nuclear non-proliferation, it's out there.
Yeah.
So, what now? Besides staying awake at night.
I don't know. I mean, I wish this is a question I'm very familiar with and do think about a fair bit and talk about with colleagues, but with no resolution, right? I'm just trying to start with the real basic of let's raise awareness so that we have people that are more informed about this and are not reacting to this. What I don't want is to be a part of some kind of movement that promotes a mass hysteria about this, right? And there are all these crazy knee-jerk reactions that aren't thoughtful, right? So are there, you know, somewhat futuristic aspects to what we talked about today that are genuinely fearful? Yes, I want to be honest about that. But knee-jerk reactions that very quickly try to immobilize those kinds of things but end up creating all sorts of other problems is not helpful in my opinion, right? We need to come together and pose this kind of question to a lot of committed and smart people who can actually do something about this from the standpoint of the regulations, right? But you raise the good point that it's not just about regulations within our country, but beyond our borders, right? It becomes that much more important to cultivate good relationships with these countries and establish some kind of an international pact. That is not going to necessarily be a panacea, right? As we have seen with many other, nuclear proliferation, right? We're still talking about it, right? And so I imagine this will be this kind of a thing, but it needs to get into that realm quickly, and we all need to convince one another at fairly high levels that this would be bad, not just for one group of people, but for all of us, right? If there were outbreaks of this kind, it would be horrendous, not just for, your targets, right? But for all of humanity. And I think an understanding of what this is and what it could really do when misused in this way is the right starting point, because it then allows us to make the case as effectively as possible. You don't want to do this either, believe me, right? And we're setting the example of how we're regulating it. Problem is going to be if we tell everybody else to regulate it and we're not regulating it, right? That's where we get into trouble, right? And we're going to create all sorts of problems. So I think we need to set the example of how we're going about regulating this, and then hope that we can lead by example there and convince everybody else with their scientific understanding of the importance of this, right? Even in those countries you mentioned, there are individuals who understand the importance of tamping down, nuclear warheads, right? And so we need to be developing those kinds of contacts and those kinds of connections with other countries. I mean, one of the lead people in Iran for this was an MIT-trained person who has really good contacts with people in the United States and in the government of the United States, and it's actually a productive interaction. And so those kinds of contacts that we can build up within the biological community will be enormously beneficial as we get nearer to these kinds of issues.
Hi, thank you so much for your talk. You mentioned a little bit about using CRISPR as it relates to diseases, and I was wondering about conversations about other differences in abilities that aren't diseases. What have those conversations been like? Because I could really see that being a very tricky ethical terrain of someone feeling like this is a problem and someone else feeling like, no, this isn't an inherent embodied problem. It's a problem of perception or a societal problem.
Right. Did everybody here understand that question? I think it's a really good question. And to some extent, it comes down to this, the nebulous concept of how to define a disease, right? Over the years, more and more things get called diseases that didn't used to be called diseases. And, if we move along with that trajectory in every kind of variation, phenotypic, behavioral variation among human beings is a disease of some kind, then everything falls in that category and we're okay with it with CRISPR-Cas9? I don't think so, right? That I may be prone to anger, I don't know if I'm not, but if I am, but that's a possibility because of my genetic predisposition together with my environmental variables and all of those complexities, is not the same as someone having metastatic lung cancer. But those are extremes on a spectrum. And we can all kind of shake our head and go, yeah, I get that, right? It's what you're talking about is this stuff in the middle as we deal with the no here and the yes here, and then we get closer to the middle and we go, what do we do with all this, right? I actually think that in most instances, there's a clean scientific line that we can draw. And what I mean is this. Most complex traits, and that's what I would refer to, that'd be the umbrella category for the vast majority of the kinds of things you're talking about, right? Like traits of human beings, right? The vast majority of complex traits, they're called complex in the scientific community because there are many, many, many variants or mutations across the genome that contribute each a tiny bit toward your predisposition toward those traits. Right? So I'll give you kind of a prosaic one, which is height, right? And maybe if you want to get taller or shorter, or I don't know why you want to get shorter, but if you want to get taller, I want to get a little bit taller, right? There are hundreds to thousands of loci, and that's probably an underestimate, across our genomes that contribute to our overall height. And that's just the genetic component. Then, of course, there are all sorts of environmental components, right? And so that's not something we can realistically really touch with CRISPR, right? Because then you're just, at that point, you're just making a new genome, right? Which is a whole other kind of issue and question. With CRISPR-Cas9, I think that the greatest power of this is going to be limited to cases where we're able to, it boils down to 1 mutation, 2 mutations, 3 mutations, a handful of mutations that we're going to, it's manageable for us to be able to correct all of those in the same cells. When you start to get to 5,000 regions that you want to correct in order to influence your height, I think we can draw a line there. And maybe it's a line that we keep revisiting because the technology gets better and we can do 5 and we can do 10 or something, I don't know. But right now, we're not at a place where we're even thinking about stuff like that because it's just not realistic scientifically. And the technology, we're not in a position to really be able to do that effectively. But maybe we will. And then your question is going to become even more relevant. My encouragement would be to try to set that aside for the moment, because I think it's going to, I think it's going to muddy the waters in the way that we think about CRISPR-Cas9 for diseases. I think if we define the sliver of things that we're comfortable doing fairly conservatively, right, and start there, then we're positioned to be able to ask these kinds of questions. Does that help at all, or? Yeah, okay.
So we have one over here, and I think we have time for about 2 more.
So thanks again for a great talk and for making both the technical and the ethical very accessible. I'm wondering about, in terms of somatic mutation, alteration, how does CRISPR technology, and maybe in the case of HCM, how does that compare with pre-implantation genetic diagnosis? And
I'm sorry, what was the last thing you said?
with pre-implantation genetic diagnosis, because I'm not clear exactly. I understand that, in one you're manipulating the genome and the other you're just choosing.
Right.
But if you're correcting HCM in an embryo, does that, are you just changing it from the deleterious mutation to the wild type, and therefore is it that embryo then identical to one that would have been chosen in pre-implantation genetic diagnosis, or are there any other differences there to be aware of, and that would inform an ethical decision that is distinct from the ethical decision to do pre-implantation genetic diagnosis?
Yeah, I mean, I don't know enough about what happens, to be honest with you, about the pre-implantation genetic diagnosis and the use of that in developing a strategy for the individual. But it is the case, ideally speaking, that in this genome editing correction version of it, that you are only making a change in that area, right? In that particular mutation that we know to cause the disease is being reverted back to something else that doesn't cause the disease, which we have mapped out fairly well, right? So in an ideal world, that should be the only thing that's happening, right? I did bring up that we are still at a place where off-target effects is a real topic, and so it could very well be, and I don't want to get into the details of this, but if particular regions of the genome are harder to tackle without off-target effects than other parts of the genome. And so if the mutation we're trying to correct happens to be in one of these sticky parts of the genome that are a little bit harder to do more specifically, in other words, there might be more off-target effects, then that ideal scenario is disrupted, and we have to deal with the ethical consideration of having off-target effects. But outside of that, the idea is exactly this, that you would just be correcting that mutation.
Go ahead, yeah.
Did you have a question? Okay.
Again, thank you very much for your really bright talk and all the questions that you raised. And I'm on the heels of previous question, because as a scientist, we all know that there is no truly ideal method. And so you talk about the method CRISPR-Cas9 as mostly ideal method. Can you, from a point of view of just lab work, real research, because it's needed for us to know, really outline the problems that you see even with implication of methods right now in a lab? What potential problems that you see?
Technical problems?
Yes.
Yeah, so one that we've been talking about a lot, which I won't belabor that point since I've mentioned it, is this issue of off-target effects, right? So that's one. And does everybody understand kind of what that means, right? You're not being as specific as you'd like. For various reasons, you end up hitting other regions that you didn't intend to, right? So this is a serious concern. The second would be something called mosaicism, right? And so this is a fancy word for having a patchy effect, right? So if you've got a cluster of cells that you want to make this impact in, you want to make this edit in, right? With each of those cells having a genome that you want to then go and edit, right? We're still optimizing the technology to be able to do that at 100%, right? So what if we go in there and only get 80% of the cells, right? Is there a detriment to doing this repeatedly so that we get everything? Is there a problem with going through with this and then leaving 20% and it's the patient still having some risk? So mosaicism is still an issue. That would probably be one of the major technical hurdles that people are trying to work on. Both of those have improved dramatically, even in the span of the last 5 years. Right? So it's been extremely encouraging and heartening to see that improvement. But I don't think anybody will, I can't speak for everybody, but I think very few people will probably get up here and say, we're done. We're ready to go with this. Right? So I think those would be the two most salient ones. Yeah. Thank you. And as I said, the other issue is just, we've come across this in our research. I mean, not from a clinical perspective, but to some extent, it's dealing with the same genome. Some parts of the genome are just a lot more difficult to attack in this way. It's not one size fits all, right? You need to really design the machinery, custom design it for your location of the genome of interest. And some locations of the genome are more refractory to this than others. And that impacts not only specificity, right, off-target effects that I was pointing out, but also potency of effect at your location of interest, right? And that may be really difficult hurdle to overcome for some locations of the genome, right? And this is something we always end up finding. We rush headlong into this because we're excited. Oh, the neighboring lab did it for one location, and I'm going to do it for mine. And then we realize that our location comes with all sorts of baggage.
So we're at close to time. Kermit, did you have a quick question? Here we go. Here we go. Just
hopefully the question's quick. The answer, who knows? The short form of the question is, it's so easy, so quick. It's kind of, in a way, piggybacking on Ed's. What about biohacking? What about,
what about what? I'm sorry?
Biohacking, basement, hey, I'm going to do some work. I saw this on YouTube. What's the thinking there? Or is that a potentially big problem or not?
Yeah, this is, when I say that it's easy to use and it's fast and it's cheap, it does raise these kinds of concerns, but it still requires some technical knowledge, right? It's not like anybody can, this is now like a full democratization and anybody and everybody can go do CRISPR-Cas9 now. It was more in the context of, okay, it's gotten easier than other technologies. It's gotten better than other technologies. But as things get easier and easier, that's the trajectory we're dealing with here. It's not a totally out there question, right? And that's sort of what I meant by getting it in, having it end up in bad hands, and people that have nefarious motivations, right? There's still going to have to be people that are they're well connected somehow, whether it's black market or above ground. And still have technical knowledge to be able to execute on this and to be able to troubleshoot. Every single CRISPR-Cas9 experiment we've attempted has not gone perfectly the first time we went through with all the reagents we designed. We needed to go back, optimize some things, come back to it. All of that requires space to do this work and money. Even though it is getting cheaper, it still requires money. Right, resources that need to be plenty in order to continue this. So it still requires a certain amount of knowledge, facility, and access to pull it off. So it's not like anybody sitting in their basement can now do this or something.
Let's thank Praveen.
Thank you very much.